Today, we are going to discuss RNA isolation. We'll first discuss theory behind how RNA isolation works, and then we will demonstrate the steps it takes to isolate high-quality RNA. Let's discuss the background behind RNA extraction first. RNA extraction is the purification of RNA from biological samples. Unlike DNA, RNA is more unstable due to the presence of a hydroxyl group at the 2' position of the pentose ring. To further complicate matters, the ubiquitous presence of ribonucleases in cells, tissues, and environment can rapidly degrade RNA, thereby complicating the RNA extraction process. To prevent this, any equipment used for RNA extraction has to be thoroughly cleaned with chemicals to destroy RNases. In addition, experimenters have to take special care to ensure their own skin RNases do not contaminate their own samples. There are several methods to extract RNA from samples, but the most commonly used method is guanidinium thiocyanate phenol chloroform extraction, or more commonly known as trizol extraction. This method is a liquid-to-liquid extraction method, which may take longer than a silica column-based method, but has a higher recovery of RNA. This technique works because it relies on phase separation by centrifugation of an aqueous sample mixed with a solution of phenol and chloroform. This results in the formation of an upper aqueous layer and lower organic layer, consisting mostly of phenol. Guanidinium thiocyanate is a chaotropic agent that is one of the components of trizol and is important in denaturing proteins that may bind to nucleic acids or degrade RNA. The pH for RNA extraction is extremely important because low pH, around 4, ensures that only RNA preferentially remains in the aqueous phase, while DNA and proteins remain behind in the organic layer. In contrast, for DNA extraction, the neutralization step, which brings the pH up to 6 or 7, allows both DNA and RNA to partition into the aqueous phase. So, under low pH conditions, we can isolate the aqueous layer containing RNA. Isopropyl alcohol can then be added to this aqueous layer to precipitate RNA. The isopropyl alcohol disrupts the hydration shells, neutralizing the negative phosphate backbone of RNA. Instead of water molecules, positively charged metal salt ions neutralize the negatively charged RNA. The neutral RNA begins to aggregate in the aqueous solution, following which the precipitated RNA is pelleted after high-speed centrifugation. A subsequent wash step with 70% ethanol is used to remove salts and concentrate the RNA. Finally, the use of depth sea water is used to redissolve the RNA pellet. Before starting, make sure your working area is cleaned with RNA Zap. The first step is the homogenization step. Pellet all the bacterial cells growing overnight by centrifuging at 12,000 Gs for 2 minutes. After centrifugation, the Luria broth supernatant is then decanted. The pellet is resuspended in trizol. lyse cells by vigorously pipetting a few times. Centrifuge sample at 12,000 G for 10 minutes at 4 degrees Celsius. All nucleic acids and proteins in the supernatant are transferred to a new tube. Take special care because phenol inside trizol can cause the microfuge lid to loosen. Do not leave the microfuge tube containing trizol inverted or laid down sideways, as the solution may leak. Next is the phase separation step. Chloroform is added to the sample. The sample is shaken vigorously and incubated for 3 minutes at room temperature. After that, the sample is centrifuged at 12,000 G for 15 minutes. By the end of the centrifugation, you should be able to see an upper colorless aqueous phase, an interphase, and lower red organic phase. The RNA in the aqueous layer is carefully removed and transferred to a new tube. The next step is the RNA precipitation step. 100% isopropyl alcohol is added and incubated for 10 minutes. Then there is another centrifuge step at 12,000 G for 10 minutes at 4 degrees Celsius. The final step is the RNA wash step. The supernatant is removed and the pellet is washed with 75% ethanol. The microfuge tube is then centrifuged at 7,500 G for 5 minutes at 4 degrees Celsius. The ethanol supernatant is decanted and any residual ethanol may be removed via pipetting. The pellet is air dried for 10 minutes and then resuspended in depth sea water. This concludes our tutorial on RNA isolation. We hope you found this video informative. Thank you for listening.